very much, and thanks for the organization. Do you hear me? Is it okay? Thanks the organizer to invite me. So, as Boris has no financial relationships to disclose, I'm a basic scientist. So, in our lab, we have interest to demonstrate that myofibroblasts can have some interaction with other cells in the tissue. And to demonstrate this interaction, we are using tissue engineering method. This method is very interesting because we can use in vitro cells, but these cells are on similar conditions than in vivo. Okay, so Boris have made a very nice presentation of myofibroblasts, so I just skip quickly on this slide. But the important thing that you have to know is myofibroblasts are usually not present in normal skins, but they appear during the normal wound healing, and they are present in pathological tissue as hypertrophic scarring. So our main interest was from interaction, and at the beginning we have thought that keratinocytes, endothelial cells, and myofibroblasts appear at the same time in the wound. So we hypothesize that interaction occurs between the cells during normal healing, but also during pathological fibrosis. So first of all, we have been interested by interaction between epidermal and dermal cells. So to understand this interaction, we have used a self-assembly, a new model that we have developed in our lab. And it's a tissue engineering approach, and briefly, dermal cells were cultured with ascorbats, and they made their own matrix as in vivo. And we can have and obtain manipulatable sheets that we can superimpose to obtain a thicker dermis. Keratinocytes were then added on this dermis, and when they reach confluency, we can elevate the skin at the air liquid interface that they can induce the maturation of the epidermis. So to understand what are the role of myofibroblasts and keratinocytes, we have used two types of population of myofibroblasts. The first one was isolated from hypertrophic scar myofibroblasts, from hypertrophic scar, sorry, and the second was from normal wounds. And for sure, you have used control with normal fibroblasts. Epidermis was made with hypertrophic scar keratinocyte or from normal skin keratinocyte. And this is an example of what we obtain using trichromason histology. And you can see in blue the dermal parts, and in red the epidermal part. And we can see here, when we add a hypertrophic scar keratinocyte, we have obtained a dermal that is thicker than when we use normal keratinocyte. Of course, we have measured the dermal thickness of this, and when as we can see here, independently of the dermal cells, we have obtained a dermis that is thicker when we use hypertrophic scar keratinocytes than when we use normal keratinocyte. We have then tried to understand what are the specificity of the hypertrophic scar keratinocyte secretions. So we have used two-dimensional electrophoresis methods, and we have used a supernatant of culture keratinocyte, and we have detected that when you use keratinocytes that were isolated from hypertrophic scar, we have obtained a very constant and very important spots that they are never or weakly present when we use normal keratinocyte supernatants. We have isolated and sequenced these spots, and these spots contain mainly TIMP1. What is TIMP1? It's tissue inhibitor metalloproteinase 1. And of course, it inhibits MMPs or collagenase, but it has been also shown to stimulate cell growth or inhibits apoptosis. We have measured in the supernatants using ELISA methods the presence of TIMP1, And as you can see here, in monolayer culture keratinocytes, supernatant, although in skin equivalent supernatant, we have obtained an increase of TIMP1 secretion when keratinocytes from hypertrophic scarring are used. We have also added TIMP1s during the formation of the dermis, and as you see here in green, you can see that when we add TIMP1 in skin realized with normal keratinocytes, we obtain a dermis that is increased and is very similar than when we use hypertrophic scar keratinocyte. So we have validated our finding using biopsies that we have obtained from hypertrophic scarring or from normal skin. And as you can see here, in the epidermis, you can see that the keratinocytes secrete or produce more TIMP1 than in hypertrophic scarring than in normal skin. So usually hypertrophic scar are thought to be mainly a dermal pathology. And as you can see, using tissue engineering method, we have detected that keratinocytes play a significant role during fibrosis formations. So the second, we have studied the interactions that can be played between endothelial and dermal cells. Again, we have used tissue engineering methods. This time we have used a fibril gel. That's just a model of the first time during the healing. And again, we have used two different populations of myofibroblasts, normal fibroblasts or hypertrophic scar myofibroblasts, that we have mixed with endothelial cell isolated from normal skin. We have put both cells on fibrogene, and after 40 days, we have evaluated the network of angiogenesis. This is the morphological aspect of fibril gel. As you can see here, when you add only endothelial cells, we cannot, we can detect a weak network of a capillary-like network, but when we add mesenchymal cells, we can see a very strong network. We have studied the network using a collagen 4 antibody, and we can see here the result with only endothelial cell, and the three images is endothelial cell and mesenchymal cells. And we have measured this result, and we have calculated an angiogenic index. It's a reflection of the network. And as you can see, when you add here in blue is the result with only endothelial cells. In gray, it's endothelial cell with fibroblast, and in red with myofibroblasts, and in yellow with hypertrophic scar myofibroblast. And you can see here that in presence of fibroblasts or myofibroblast, we have obtained an increase of the angiogenic index. But when you add normal myofibroblasts, this increase is very important and significant. This is 3 different experiments that we have obtained with different populations. So during wound healing, so endothelial cells and myofibroblasts can interact to form capillary structure. Furthermore, myofibroblasts sustain capillary formations better than non-differentiated fibroblasts. It was known that fibroblasts can help capillary formation, but it's very new that myofibroblasts can have a role during the angiogenesis. Usually, myofibroblasts are thought to have a very important role during the matrix formation or during the contraction of the wound, but angiogenesis formation is very new. So my final conclusion was that interactions between numerous cell populations contribute to normal and pathological tissue formation, and these parameters have to be thought when we study this different pathology. Thank you very much.